So I'm going to be discussing the proteins involved in VDJ recombination, and I thought to start out with I'd give a little overview of what is VDJ recombination. So basically what's happening is DNA is encoding a complete antibody V region, and it's going to be assembled from the V, D, and J of the heavy chain or using V and J of the light chain. And these two segments are initially going to start out being many kilobases apart. And you're going to have each developing new B cell is going to generate a new pair of a variable region combining each of these segments, and it's going to be recombining at the genomic level of DNA. And this whole recombination process is actually directed by a series of enzymes or proteins. And recombination is actually going to be directed to the appropriate sites on the immunoglobulin gene, and how it's going to recognize that is there's going to be specific DNA sequence motifs, constant patterns that reoccur called recombination signal sequences or RSSs. You'll hear that referenced a lot. And these RSSs actually ensure that one of each type of the segment, the V, D, and J of the heavy chain, or the V and J of that light chain are included in the newly recombined varied gene region. So basically, overview of what's happening in that recombination is these RSSs are going to go through basically a recognition, a cleavage, and then going to be ligated back together. And so during all this cleavage and ligating, all this editing is basically just creating diversity and adding to the further variability of that recombined gene. And so now I want to discuss specific proteins and enzymes that are aiding in this whole recombination mechanism. And I thought the best way to do that would be to basically break it down, kind of simplify it step by step, and show you which proteins are doing what at which step of this recombination. So to begin with, there's a very important protein known as RAG1 and 2. RAG stands for recombination activating gene, and this protein and protein complex, they're actually closely linked, encoded by closely linked genes, is going to be responsible for the very first major phase that's happening in recombination, which is going to be recognition and then cleavage. And so what's happening actually is RAG1 and 2, this complex, are going to be recognizing that RSS we talked about earlier. And after recognition of the RSS sequence, then the RAG complex, RAG1 and 2, are actually going to perform single-strand cleavage. So basically what they're going to do is a single-strand nick on that coding strand of each of the V segments, and they're also going to do a single-stranded cut on the non-coding strand of the other segment. And some important things to note about RAG1 and 2 is that RAG1 is actually our predominant recombinase, because it's going to actually be forming the complex with that recombination signal sequence. And RAG2 is basically there to stabilize the binding of RAG of the RSS. And it's also been found that RAG2 by itself does not exhibit any RSS binding activity, and that's kind of what shows us that RAG1 is the predominant recombinase acting here. Important to note that RAG1 and 2, along with another protein called TDT, are actually the only 3 proteins that are lymphocyte specific, meaning they're actually unique to our lymphocytes. And they're the only 3 proteins involved in the recombination that exhibit that characteristic. And so now with our RAG1 and 2 complex, we also have a few other proteins helping it called the HMG1 and 2 proteins. And these proteins are basically just helping to stabilize the binding of RAG1 to that RSS sequence we talked about. And so now in our recombination process, we've been recognized and cleavage has happened. So the next step is basically V and J region hairpins are going to form, and the end of these hairpins are going to have very blunt, sharp signal ends. And so you got these ends and an enzyme called DNA ligase 4, which you're probably familiar with, is going to basically come in and ligate or tie up those free blunt ends to form a signal joint. Now, this DNA ligase 4 is also going to have some help for other non-specific lymphocyte proteins, and these proteins are known as XRCC4 and Cernunnos. Sorry, that's a hard word to say. And those two proteins are basically going to help in activating and stabilizing that DNA ligase 4. And so our next step after that's happened is basically our hairpins at the ends of the V and J regions are now going to be opened up. And so there's a protein known as Artemis that is actually going to be responsible for catalyzing the opening up of these hairpins. Important thing to know is that the Artemis protein is actually opening up the coding ends of these hairpins. And Artemis is actually going to have some help from some other two non-lymphocyte specific proteins known as the Ku70 and Ku80 heterodimers and DNA PKCs. And basically these PKCs and those heterodimers are going to complex together, and they're going to help with the recruitment of Artemis and the phosphorylation of Artemis. And our next step after that occurs, the opening of the hairpins, includes something called overhand extension, and that's going to be followed up by further ligation of that light chain. And of course, ligation is predominantly done by that enzyme DNA ligase 4. The next step after that ligation is something called exonuclease trimming, and right now they're not actually 100% sure which protein is responsible for this exonuclease activity. They have two guesses that it could either be the RAG proteins or Artemis protein. The reason they think it's the RAG protein is because they already know that the RAG protein itself can actually trim DNA that's near a 3' flap, and they just have seen that Artemis actually has both exonuclease and endonuclease activity, so they think that it could also perhaps be responsible for the exonuclease activity that's being undergone in the VDJ recombination mechanism. So after exonuclease activity occurs, then we have something called N-nucleotide addition. And this N basically just stands for a non-templated, untemplated nucleotide. And these are going to be added to that coding joint of the heavy chain genes by a protein known as TDT that I kind of mentioned earlier, because it's actually one of the proteins involved that are lymphocyte specific. TDT stands for terminal deoxynucleotidyl transferase. That's a mouthful. How it's going to work is actually it's going to add nucleotides to that free 3' termini of the coding ends. And TDT, can actually add up to 20 nucleotides at a time on each side of that joint. And it also actually helps function in holding the two together. Another thing that TDT can do is it's actually responsible for the generation of adding some extra diversity to the CDR3 region of the heavy chain of the antibody. So some important things it does. And after the TDTs come in, we're going to have the very final step, basically sealing everything back up. Ligation, of course, is going to be done predominantly by DNA or ligase 4. And so now I have a very detailed step-by-step diagram of what's happening in this recombination I wanted to show you and point out the proteins that we discussed as they come up. So like I said, in the very beginning, we're going to have, can you see that? Yeah. We're going to have RAG1 and 2 at work with those proteins that are going to help it out, the HMG proteins. And those are going to start out by recognizing that RSS sequence and then cutting the DNA. It's going to tell you in step 2 that it performs that single-stranded nick. That's important to point out. And you're going to see afterwards come down here to step 4, the ligation that occurs of those signal ends we talked about, the sharp ends that were cut off. And of course the ligation is going to be occurring via primarily DNA ligase 4. And after ligation occurs, we're going to come up here to step 5. I talked about the overhang that occurred, and the opening of the hairpin that will result in overhang. And we talked about what the protein was that was performing the opening of this hairpin, and that was the protein known as Artemis, of course. And after Artemis, we're going to have more hairpin cleavage, and then it's going to be followed by, of course, ligation down here, and that's going to be done by ligase 4, DNA ligase 4. And so then we're going to come over here to step 8, which we talked about earlier, that exonuclease activity, which they said they weren't 100% sure right now which one it was, the RAG proteins or that Artemis protein. It could be either or, but after exonuclease activity, of course, they're going to be, we're going to have the protein TDT that's going to come in, and it's going to add those N nucleotides we talked about. N stands for non-templated nucleotides, and TDT is going to come in and join those up. And our very last and final step, step 10, ligation of that heavy chain by DNA is going to occur via ligase, DNA ligase 4. And so, the last thing I kind of want to talk about is how can we relate this back to our everyday lives, or what can we get out of this? Well, the whole process, VDJ recombination, is meant to generate diversity among our T-cell receptors. And it's a very important mechanism that is contributing to the diversity of antibodies in our immune systems. And the whole mechanism that results in so much cutting and rejoining of parts is what's actually producing all this diversity and contributing significantly to the variability of our genes. And this is important because we want our immune systems to have incredibly diverse antibodies and our genome to be always diversifying. That way our bodies can be able to, and our immune systems can be able to adapt to anything and figure out a way to respond to any new antigen that's thrown at it and adapt to it. So that's all I have about proteins in the VDJ recombination mechanism, and feel free to use my video. Thanks.